Since 1981, the HIV/AIDS pandemic has killed an estimated 25 million people worldwide. Although mortality rates have declined since the 1980s, especially in developed countries, the disease still claims millions of lives every year. In 2005 alone, it is estimated that between 2.4 and 3.3 million people died of AIDS, the advanced stage of HIV.
Approximately 33 million people are living with HIV virus, and then over 1 million people in the United States are infected with this virus.
Although incurable, meaning it cannot be eradicated from the human body, HIV can be controlled by drugs.
Currently, the treatment of the HIV infection heavily relies on what we call HAART therapy, referred as to highly active antiretroviral therapy.
But there are problems with these drugs. Most of them have strong side effects. Roughly a quarter of all patients stop therapy within the first year due to the effects of high toxicity. This obstacle motivates scientists all over the world to discover better treatments for HIV and AIDS. One of these researchers is Sha Jin, assistant professor of biomedical engineering at the University of Arkansas. In her lab at the Engineering Research Center in South Fayetteville, Jin works with HeLa cells to develop a molecular probe that can simultaneously detect HIV-related enzyme produced by those infected with HIV, as well as toxicity levels of compounds used in HIV drugs.
We tested the tube developed in my lab, and we found that if we add HIV inhibitor to the cells, we receive higher signaling, and because cells showing us red or yellow color, that indicating the HIV protease activity was inhibited by its inhibitor.
And so what Sha Jin in biomedical engineering has done is something I think is rather clever. She knows the protein sequence that the HIV protease will recognize and clip. What she's done is to take two proteins that will actually respond to light, but when they are in close proximity to each other, they'll behave in one way, and when they are not in close proximity to each other, they'll behave in a very different way, and you can see that. What she's done is to take the protein sequence that this HIV-1 protease attacks and essentially join these two light-responsive proteins. And she can express in HeLa cells this light-responsive protein combo along with the expression of the HIV-1 protease. The protease is active when it's expressed, and you know this because it goes and clips the target sequence that she put in between the two light-responsive proteins.
Basically, we're using DNA cloning technique to create a new probe, which is able to tell us which chemical compound has anti-HIV activity.
Jin genetically engineered a biological screening system by applying fluorescent proteins, one green and one red, to generate what is known as Forster resonance energy transfer signals, or FRET. FRET is a mechanism of energy transfer between a donor and receptor chromophore, which is the part of a molecule that is responsible for its color. This process, the energy transfer, prompts reactions from cells in the form of color.
Using the cell, we will see the cell show in yellow or green color, indicating the lower signal because of the cleavage by the protease. So the two proteins separate apart. The distance definitely longer than 10 nanometer, then there's no that strong signal.
In Jin's project, FRET signals responded to the presence of HIV protease, which is a family of enzymes or proteins that are essential for the life cycle of HIV. The signals also responded to various compounds that are designed to block or inhibit HIV protease. When applying the fluorescent proteins to create FRET signals, Jin observed green and yellow cells in the absence of protease inhibitors. She saw red when the protease inhibitors were added to the cells. The red cells indicated HIV protease loses its activity due to the presence of protease inhibitors in the cell culture medium. Meanwhile, healthy cells growing on culture plates allowed her to measure toxicity levels, that is, the extent to which toxic inhibitors affected healthy cells.
One of the clever things that Dr. Jin has done is she's created a platform, a cell line, that gives you a visible signal when this HIV protease is working and when it's not. So not only is she able to use these cells in screening drugs that are currently being developed, but even developing new drugs that we haven't even looked at. So this same cell line, it's a specialized cell line of HeLa. It's not just a normal HeLa cell. It's now a HeLa cell that's expressing an HIV protease, and it's expressing a construct that's sensitive to that protease. The point is, that cell line may be valuable for the next 10 years in coming up with a number of new drugs that could be useful in attacking HIV.
Jin's novel detection system confirmed previous studies showing that compounds nalfinavir and lopinavir are toxic to cells, since many healthy cells died after only 2 days in incubation. Indinavir and ritonavir showed less toxicity. With HeLa, Jin is establishing a cell line that will allow her to consistently express the detection system. HeLa cells are ideal for this for several reasons. They grow rapidly and can be handled in a straightforward manner. They grow in large quantity and can be grown in cell culture at much less expense than many other culture cells. And finally, targeted genes can be easily delivered to HeLa cells.
But Sha Jin's approach of having a viral protein attacking something that's visible and changing its visibility, that whole concept, I think she will continue to expand on to impact drug development research that goes beyond just HIV. That whole platform has tremendous potential.